So, just a reminder before we get started, no labs this week, as was said before, but the lab room will be open this afternoon from 12 to 5 if you are truly desperate and are trying to get your insect collection done at the very last minute. I had a collection once where I was putting the labels on in the car as I was driven to the building. So, those things happen. Learn from my mistakes. Do not repeat my mistakes. All right. And it is due today at 5 p.m. You can turn it in to me at the office or in the lab room. If I'm not around, you can just leave it. The reason I say turn it in to me is that I can put it in my back room where it's going to be locked up and not subject to people poking at it, potentially taking things, which shouldn't happen, but sometimes happens with insect collections. So, plus that way I can look over it, and if there's any glaring mistakes that you should fix before you turn it in, I can point them out to you, and you can hopefully save yourself some points. Reminder, the final is 11 to 1 p.m. in this room on Monday, so it's our usual time, just an hour longer, and the final will be semi-cumulative. 3/4 to 4/5 of material from the last exam, with 1/4 to 1/5 being big picture topics from the past 2 exams. So I'd say take a look at those past exams. That would probably be the best way to get an idea of the sorts of materials.
Is it going to be about the same length as a normal test?
About the same length, maybe a little bit longer. It shouldn't take you the full 2 hours. But yeah, that's the idea. There is a study guide posted. It's the study guide for just the new material, so you should have got an email about that.
Was this in all multiple choice, or are there going to be written portions as well?
There will be some written portions. It'll be mostly multiple choice. So it'll be similar in format to the last 2 exams, that there will probably be mostly multiple choice and maybe matching, maybe fill in the blank type stuff, and then maybe somewhere between 5 and 7 essays that will be variable from really short to, a paragraph or 2. That being said, bring a Scantron. So, Scantron required.
Do we have to bring a calculator?
I don't think so, but never hurts to have a calculator. I carry a calculator with me, so, well, admittedly on my phone. But I did carry around a calculator before my phone had a calculator, because I predate that a little bit, just a little bit. Where I grew up, there was no cell phone coverage, so even when there were cell phones, it didn't matter. Nobody had cell phones. All right.
But they were this big and they had the cord.
I'm not that old. I'm not that bad, but I did grow up in the generation of the Nokia brick phones that, 1, never died, and 2, you could put in a sock and hit somebody with. And it was a viable form of self-defense. So, anyway, Nokia phones aside, the last topic I wanted to cover was, so on Monday we covered resistance, and resistance can be kind of bleak in the sense that it kind of gives you the impression that all pest control is doomed to fail eventually. So I wanted to have a little bit of a lecture on what are the things we can do to help stop resistance. How can we get more out of our technologies? So just as a review to resistance, resistance is caused by any artificial selection of individuals that can survive the management tactic, right? So when we spray a pesticide, we are artificially favoring those pests that can survive the tactic. They survive, they reproduce, pass on their resistant genes to the next generation. So in the next gen, we have more insects that have resistance than we did before. And potentially, if we say have 2 insects that have different resistance traits, and they mate with one another, their offspring will have both of those resistance traits. So we potentially are creating super insects that are even more resistant or are resistant to multiple things. So essentially, because anytime we kill insects, we favor those that have resistance to that technique. Resistance is generally believed to evolve in any tactic that has significant selection pressure. So generally, anything that kills insects, eventually we believe insects will evolve resistance to it, whether that's rotation, biocontrol, all those things. So when we talk about managing resistance, what we're typically talking about isn't how do we stop resistance. It's a question of how quickly will resistance evolve and how much can we slow it down. So how long can we extend the lifespan of this technology before it's completely useless? Because thus far, no one's found a solution that makes these things 100% invincible to insect evolution. All right, so as I said, managing resistance is mostly about anticipating where resistance will evolve and then figuring out ways to slow it down. So how do we actually slow down resistance? What would be the ways we do it? Well, as it works out, the two ways we can do it basically involve trying to manipulate those evolutionary pressures that the insects go through. And so there's two basic approaches that actually look like they're distinct opposites of one another right off the bat. The first thing we can do is we can reduce the selection pressure that favors the resistant insects. So we talked about this a little bit on Monday, just the idea that if you're killing, say, 99% of all of the susceptible insects, you're not leaving a whole lot of susceptibles behind to repopulate the pests next time, right? So you're leaving a high proportion of resistance compared to the susceptibles. Whereas if you do a spray that kills 70%, you're leaving a much larger susceptible population behind. And they will mate with the resistant individuals, and they will produce offspring that will at least be somewhat susceptible. And so essentially, reducing selection pressure favors susceptibles, keeps them in the environment, and dilutes out the resistance genes for the next generation. So you've got a couple of ways we do this that we'll go into detail, but mostly it involves either relieving the amount of selection pressure directly by using different techniques, using different insecticides, or directly avoiding killing these insects by leaving little patches of insects that you don't treat, where the susceptibles will thrive. Then the flip side of this is you want to increase selection pressure. So the idea here is, essentially, and we'll go into this in more detail later, that there are some insects that aren't fully resistant. They may have only partial resistance, and when partially resistant insects mate with other partially resistant insects, maybe you'll come out with offspring that are 100% resistant. And so what we can do is use a high dose where basically we just nuke the field, and in that way we knock out all the partially resistant insects and they can't mate with one another. So again, we'll come back to that. So these are very different ways to approach this problem, and the specific one you use is going to be dependent on the system and the technology you're using. All right, so reducing selection pressure. Again, the basic idea here is the lower the mortality inflicted, the more susceptible insects survive, and the more of them there will be to mate with the resistant insects, diluting out the resistance genes.
If you're not spraying a certain insecticide or mode of action, will the bugs actually lose that resistance over time or no?
That is a great question. That's
If it's not affecting them, you would think they'd stop having it.
So it's a really good question. So that's something I didn't include in this, but that is a big part of resistance theory. So the idea, right, is that these insecticides target very specific parts of the insect that have essentially evolved over millions of years to be highly efficient. And so by developing resistance, what most of these insects are doing is they're modifying those targets. And what they do when they modify them is those targets become less efficient. It makes their nerves fire less efficiently, but they don't get killed by this insecticide. And if you're more likely to get killed by the insecticide than by having defective nerves, then you're going to be favored because that insecticide's around. But so we call that a fitness trade-off, that you lose the effect. Basically, you lose the best functioning nerves in exchange for being able to survive insecticides. But you're right, once you remove those insecticides, all of a sudden you've got a whole bunch of susceptible insects running around that have nerves that fire just a hint faster than yours do. And maybe they're better at escaping predators, maybe they're better at attracting mates, and all of a sudden they're mating and reproducing better than you are. And so those resistance traits tend to decrease once you remove the insecticide. And so there is this question, if we say got rid of all organophosphates, would insects eventually return to susceptible levels and we could cycle organophosphates back in and start treating them again with that technology? So that's a long answer to a short question, But yeah, that's a theory, and at this point, I don't think we've had any insecticides off the market long enough to see if that would happen. Did that answer it?
Yeah.
Excellent. All right, so how do we actually reduce selection pressure? Well, the number one way is really just sort of good old-fashioned IPM. It's diversifying your control tactics. And so basically, the idea here is you don't want to rely 100% on your chemical. So if you have control tactics other than your chemical to use, if you have viable biocontrol, if you've got a crop rotation you can use, if you have resistant plants, use those along with your chemistry. And that way, essentially, instead of putting one very strong selection pressure on the population, you're putting lots of small selection pressures. I think Dr. Shrestha uses the many tiny hammers, right? I think I saw a ton of those definitions on the exam last time. But it's that idea, that essentially you're using lots of small hammers, and that, you know, if one of those hammers fails, such as your insecticide, then you're not completely up the creek. You know, you've got all sorts of other controls to fall back on that will hopefully help protect you. And so, essentially the idea is that resistance genes are rare in the population. We're usually talking about thousandths of a percentage of insects have a resistance gene. And so, it's unlikely that they carry a gene that gives them resistance to both the insecticide and to crop rotation. And so by using, say, an insecticide and then rotating your crop, hopefully your crop rotation will kill all the resistant insects that you accidentally selected for, right? So alternatively, yes?
Is there any data to show whether the gene is inherited from the mother or father?
Most of the genes that cause resistance to insecticides are autosomal, so they are in the parts of the genome that are from both mothers and fathers. So generally, there's not a tendency for mothers or fathers to, as far as I can tell, there haven't been any cases of sex-specific resistance. But insects have a lot of dimorphism, so it's possible that if you have resistance based on, say, your morphology or your size or the thickness of your cuticle, that maybe males or females would naturally be better suited to that, just because males and females can be substantially different sizes, different life cycles, those sorts of things. So I wouldn't be surprised. But from a purely genetic standpoint, no. So, alternatively, whereas IPM really focuses on using lots of technologies other than chemistries, what can we do specifically in just chemical control? One of this is you can diversify the insecticides that you use. So it's the basic idea that we have in IPM. You want to use lots of different small hammers to provide control, so you're not providing a lot of selection pressure on one particular gene. But in this case, instead of using, say, biocontrol, crop rotation, and chemicals, you're using different chemistries. So what you would do is you would basically use different modes of action at different times. And with the basic idea that it's unlikely, again, that insects are carrying more than one resistance gene, and so it's really unlikely that they will have resistance to both neonics and, say, organophosphates. So one way people recommend doing this is using insecticide mixtures. So you would tank mix insecticides that have different MOAs and then apply. I don't recommend this. The general rule with tank mixtures is that while insecticides have been really broadly tested and their safety is well known, once you start mixing chemicals together, sometimes you get emergent properties that you're not expecting. There have been tests that have shown that while, say, a certain pyrethroid and a certain carbamate are pretty safe for fish or for honeybees by themselves, when you combine them, you have significantly higher toxicity from their dual modes of action. And so I don't think it's really a great idea to mix them in the tank, but it is something that you hear about. So, but again, the basic idea that since you're applying two chemistries, if the first chemistry has resistance problems, the second one will knock them out, right? However, something that should be noted is that this is not something you should be doing after you find resistance problems, because then you're basically just applying a selection pressure that's going to make your resistance problem worse. You should be doing this before there's any evidence of resistance. Alternatively, you can diversify the insecticides by putting them out in different patterns. So how you apply them, you can integrate diversity. So one thing would be using, say, a mosaic pattern of insecticide application. So let's say you have a field and you're spraying in strips. Maybe you spray the first strip with an organophosphate, you spray the next one with a carbamate, you spray the next one with a diamide. And that way, if there are any resistant insects left in a certain part of the field, when they move over to the next part, say they migrate across the field looking for mates, they'll land in an area where there's another insecticide that they encounter. Now, this is nice and all. One of the challenges and why, well, this is one. So this hasn't been widely applied. Most people don't do this mosaic spray. One reason is it's been suggested relatively recently, and so the studies have really just started, and it's a question of whether it works really well. Two, it means that you have to basically refill your spray tank every time you want to do a pass of the field, which is going to be a total pain. And so it's not super popular, not super practical. A more popular variant that is widely used everywhere is rotating your modes of action. So basically, you use a different insecticide sequentially over the growing season. So if you are doing an early season spray, you'd use one mode of action. When you come up for your mid-season spray, you would use a different mode of action. That way you never expose the same population to the same insecticide more than once a season. And so you're basically, if you have organophosphate-resistant insects, hopefully with the next spray, if you use a diamide, you're going to take them out. And the idea is, again, if you do this before you have resistance problems, you're not just going to be selecting for super resistant bugs. Hopefully you'll actually be controlling the resistance and slowing it down. All right. So those are things we can do to basically lower the selection pressure through basically manipulating the system to kill less pests. Alternatively, we could do this tactic, which is essentially conserving susceptible individuals directly. So let's say you have a technology that just kills bugs like crazy no matter what you do. Like BT corn, right? It's genetically modified corn. And BT corn, if you're not familiar, it expresses the BT toxin from Bacillus thuringiensis. It expresses it in every single part of the plant, the roots, all of the inner tissues, all of the actual corn itself, the silks, the pollen, it's everywhere in there. So anywhere the insect feeds is going to get a dose, and they are engineered to provide a dose that's roughly 4 to 5 times higher than the recommended lethal dose. So it's a very high dose system. So essentially, if you plant BT corn, you're selecting for resistant individuals very, very strongly. So what do you do in that case? How is it that you can reduce the selection pressure to make your BT corn last longer? Well, what you can do is you can intentionally build places in your system where susceptible individuals will survive. Essentially, this would be, say, applying insecticides in such a way that a certain number of susceptible individuals survive, and again, these will mate with the resistant individuals and dilute out their genes. So when we're talking about insecticide systems, we typically are talking about spraying at a lower dose. So spraying at the lowest dose recommended by the package, which will basically produce more susceptibles that get a really small dose, applying less frequently. So, trying to apply only when it looks like you're going to go over the economic injury level, using low persistence insecticides, so those that don't hang out in the environment very long, using spot applications. So if you've got an insect problem and you know there's just a couple hot spots in the field, kind of like melon aphids. Melon aphids tend to break out in really big patches in the melon. Instead of spraying all of your melons and selecting all of your insects, just spray the patches. That way you still have some susceptibles around. In the case of BT that I was talking about earlier, they use this last option. They preserve refuges of pests. So what BT corn and cotton growers are required to do is they have to have a certain proportion of their field that is planted with non-BT plants. So it's anywhere between 5 and 20%, depending on where you are and what crop it is. But it's an area where they are conventional plants. You're not allowed to spray them with insecticides. Their whole purpose is to maintain susceptible insects that will go out and mate with the resistant ones. Consequently, that system works really well. BT plants have been out for almost 20 years now, so that's a ridiculous amount of time. And they just found severe resistance in the United States about 5 years ago. So that's 15 years of a technology being highly resistant. And even with the resistance problems popping up, they're very patchy. They tend to be in areas where corn has been grown consistently year after year after year. They're not rotating it, which would be helpful. They tend to be in areas where it's kind of debatable whether the farmers are following the recommendations 100% of the time or not. So what they do now to avoid that is if you buy a bag of BT corn, it's got 5% susceptible seeds mixed in it that you can't tell the difference. So when you plant, you automatically plant 5% susceptible. All right, and then finally the high dose tactic. So the high dose tactic I explained a little bit before, right? Just the idea that most insects aren't going to be 100% resistant. It's going to be really uncommon that you're spraying an insecticide and you just so happen upon a bug that you spray it with a full dose and it doesn't die. What you tend to find is that most insects have partial resistance, or essentially they have a small amount of resistance conferred, so that if they encounter, say, a lethal dose or a slightly lethal dose, something that would kill, say, 50% of bugs, they're more likely to survive. And then we get full resistance when those individuals crossbreed with one another, and we end up with multiple resistance traits building up in a population. So high dose basically just says, let's dose this field at such a high rate that all of those partially resistant insects will die. They won't have a chance to mate with one another, and we won't end up with resistance. This is the theory that is being used in BT corn. And as I mentioned, using a high dose in mixture with conservation of susceptible individuals has been really successful in BT. And looks like it's going to be the paradigm that we use moving forward for a lot of different pests as far as getting more bang for our buck from technologies. All right. So those are the ways we get at resistance. And then finally, I've got two things that you might see in the field with insects that aren't resistance, but are major problems that could be caused by insecticides. These are situations where essentially you would apply a pesticide and then it backfires on you and you have a worse problem than you did before. So one of these is resurgence. Resurgence is when a population, after having been suppressed, rebounds in numbers greater than they were originally, right? So if here on the vertical axis we have the pest population, and then we've got time, we've got our usual sort of pest population, right? It goes up, it goes down, but it tends to be right around sort of a mean population size. Eventually, let's say you treat the population, it crashes, but then for whatever reason when it comes back, its mean is shifted. Now the average insect population is much higher than it was before, and chances are it's above the economic injury level. The other trend that we sometimes see is what's called replacement. So replacement is when you have one pest species, right, that is your big pest problem. And then you've got a second species, a secondary one. So you got a key species and a secondary species. Secondary species tends to have low pest population, key has high. Sometimes what you do is you apply a pesticide, you knock down that key species, and all of a sudden your secondary species population growth explodes. And it ends up becoming a new key pest. So basically you've switched. But now you're spraying all the time to try and keep this key species low, and you also have to spray to try and knock down the secondary pest. So you're probably doing twice as many sprays as you would have been in the first place. A really good example of this would be the Imperial Valley in California. They had a really strict regime of spraying broad-spectrum insecticides to control pink bollworm. The problem was when they did that, they knocked out the bollworm, but they also basically wiped out all of their natural biocontrol they had going on. And the biocontrol was basically protecting them from this guy, the cotton perforator, which is another moth species. And essentially, once that biocontrol was gone, cotton perforator became a major economic pest, and they had to start spraying for that too. So they ended up with a problem that was worse than what they had initially. It's not a term I put in here, but we call that the pesticide treadmill, which is the tendency that the more pesticides you use and the more reliant you are on pesticide, the more of these sorts of problems you breed. You tend to start seeing resurgence problems, replacement problems, and of course resistance problems, to the extent that you're applying so many insecticides that it starts to really destroy the economic viability of your crop. There have been systems, especially in Central and South America. We sort of exported the American way of growing crops down there, And a lot of those cotton growers down in Central America basically realized that to control all the pests they had that had evolved resistance and resurgence problems, they were actually losing money every single year on cotton just from all the sprays. There were some systems where they were spraying up to like 18 times a growing season. So we're talking very high amounts of insecticide going into them. All right, so what actually causes resurgence and replacement? Well, the first one, and the one that seems to be the most widespread, is what I talked about just a second ago. It's a reduction of natural enemies with pesticides. So essentially, with resurgence or replacement, you probably have some biocontrol agents in your field that are providing control. They are either keeping that key species a little low, or maybe they're providing suppression of that secondary species. You spray an insecticide that is broad spectrum, kills all of your biocontrol, and all of a sudden, there's nothing keeping those populations in check anymore. A big situation that's kind of famous for this was the brown plant hoppers. So I believe it was Malaysia. The government basically wanted to boost their agriculture sector, and so they subsidized insecticides 85%. So growers only had to pay 15% of the cost. And so what happened is what you would expect. Growers started spraying a lot of insecticides, and it did boost rice production for a few years, and so it was considered this big success. The problem was they essentially knocked out all of their biocontrol. Biocontrol didn't bounce back when they slowed down their spraying, and these plant hoppers, which had been a really minor pest for a long time because they had this huge biocontrol suite for them, they became a huge problem, and they started spreading a lot of viruses amongst the rice, and eventually they got to a point where they were actually losing money on rice every year. New government came into power, they got rid of the subsidies, people stopped spraying, and when the biocontrol came back, they actually had a 15% boost in yield just from biocontrol action. So this is very well studied, and it's known to have occurred a number of times. So getting rid of your biocontrol can really screw up a system, even if you don't see a whole lot of it going on. The other cause? Alternatively for this, so I talked about that in the case of a secondary pest. If you're looking at natural enemies in a resurgence case, right, where you have a pest at a low level, you spray it and it comes back at a high level, that would be a case where, say, you've got a key pest, it's being maintained at a low level by biocontrol, you spray, you kill both the pest and its biocontrol, so when the pest comes back, it reproduces faster than the biocontrol agents, and it reestablishes the new population at a higher average. Okay, so the other major cause that you typically see with resurgence and replacement is insecticide homologosis. So we talked about this in lab a few times, but just to remind you, essentially it's a case where it's a direct favorable influence on pest physiology or behavior caused by insecticide exposure. So essentially you get a sublethal dose, and the insect comes out of it better off than it was before. It either produces more eggs, maybe it's more successful at mating. Either way, you end up with a much higher pest population after the spray due to a physiological change. And this has also been widely demonstrated. It's known in spider mites, Helicobacter zea, the tomato fruit worm, brown plant hoppers, corn rootworms, all of those things. And there's actually increasing evidence that this is a really important factor in a lot of insect systems. It's just hugely understudied. And so I'll be kind of curious to see where homologosis goes from here. And again, how that's going to impact the way that we think we should apply insecticides. And as I mentioned before, it's really unclear what causes homologosis. Some people believe that the insecticides may alter the plant physiology to make them more attractive to the insects. Maybe it initiates reproductive behavior in insects. In the plant hoppers, the brown plant hoppers, they discovered that having a sublethal dose actually caused the larva to live longer. So you had an extended feeding phase, and it's spread out when they lay their eggs, so that they laid their eggs all season long instead of these quick bursts that were easy to spray for. All right, and then the final cause is the removal of competitive species. So the basic idea here is that if you have two pest species, right, you got a key species and a secondary species, the theory is that the reason the secondary species is kept low is because it's competing with the key species for a limited resource, right? There's only so much corn to eat, so maybe corn rootworm is a secondary species because Helicobacter zea is consuming all the corn that it wants to. So by getting rid of Helicobacter, all of a sudden you've removed that competition, and the corn rootworm can increase, which is kind of nice. It kind of lines up with the current paradigm of like free market economies, right? It's like competition, competition, competition. But it turns out there's really not any impressive experimental evidence that this is happening at all. And people have tried. So I'm not super hot on removal of competitive species. I think there's a lot of evidence to suggest that homologosis and reduction of natural enemies can really screw up insect systems. But competitive species, not so much. All right. Oops. Almost. There we go. So what can we do? What can we do to actually manage resurgence and replacement? Well, the first thing we can do, right, is we can avoid killing those natural enemies. And the easiest way to avoid killing natural enemies is to use selective insecticides. These are insecticides that are designed to basically kill just the pest that you intend to kill. There's two ways you can get at this. You can have physiologically selective insecticides. So these would be insecticides like the diamides. They have a mode of action that is designed to basically hurt pests more than they hurt biocontrol agents. So diamides are known to be highly toxic to a variety of pests, but they don't hurt honeybees. They don't hurt parasitic wasps. They generally are much safer for Hymenoptera than they are for, say, beetles. So this would be an example of an insecticide that you could spray that is selective for pests. Alternatively, there are things like ecological selective pesticides. These are broad-spectrum insecticides. So these are insecticides that do kill everything, but you find a way to apply them so that they only come into contact with the pest. So a good example of that would be systemics. You apply something to the roots, it gets picked up by the plant, and it's circulated inside the tissues. So in most cases, a systemic insecticide is only going to come into contact with insects that are actively feeding on the plant, which by definition are going to be pests. Sometimes you get systems where predators feeding on the insects feeding on the plant might get residuals of the insecticide as well. So you got to watch out for that. Alternatively, there are things like baits. So, putting out a food source that the pests will eat that has a toxin in it, or timing your sprays to times when the parasitoids or the predators are expected to not be out and active. Ooh. Alternatively, if you want to avoid homologosis, the best way to do this is to dose correctly. So use a high dose. So you reduce the number of chances that you're going to get those sub-lethal doses that cause them to bounce back up. And then finally, this is kind of the last-ditch approach. If you're using something that's going to kill your biocontrol agents, you can always release more biocontrol agents. You can buy biocontrol agents from any number of retailers. Don't go to your realtor. My parents were just in town last week looking at houses, so got houses on my mind. But as far as I know, from going around with Ken, he did not have any biocontrol agents at the time.
Not on hand.
Not on hand. It's probably a deal. You have to buy a house or something. They probably just don't give them away. Anyway, so you can buy biocontrol agents, and you can release them in a system where maybe you not accidentally, but maybe you killed all the biocontrol agents. You can re-release. And actually, there are now some companies that specialize in insecticide-resistant biocontrol. So you can buy a bunch of wasps that are resistant to organophosphates, and you can release those. And then the next time you spray, you don't have to feel so bad.